So I've got this interesting question here asked by some person. So they ask, so are the receptors that the neurotransmitters bind to themselves the membrane proteins? Does that mean it's fair to say the neurotransmitters change the shape of the receptors or the sodium channel proteins? Finally, can one neurotransmitter cause an inhibitory response in one synapse and an excitatory response in another? To me, it doesn't make sense because the neurotransmitter only binds to receptors which are complementary to its shape, so they will only bind to one type of receptor and one ion channel protein type. Yet again, bio never really made any sense to me, so I don't know. Well, the good thing is, some person, if you're asking questions like this, that means you're starting to really think about what's going on and how how this machinery all works. So the good thing is that even though some parts of biology don't make apparent sense, you're asking the questions, which means you've probably got a good understanding of the key points, and now you're starting to look between the details that are there. So you're in a good place to begin with. We never really fully understand everything. That's just how things work. All we're ever really doing in biology is asking the next question. And when we ask a question and we get that question addressed with an answer, it really just takes us to the next question, really. So once we get to one level of understanding, we're always asking questions about the next level of understanding, and that's really what science is all about. So the first question was, are the receptors that the neurotransmitters bind to themselves the membrane proteins? And so for A-level biology, what we will assume is that yes, the neurotransmitter. So let's just remind ourselves about what's going on. We have the pre-synaptic neuron. Okay, and it's got its synaptic vesicles near the presynaptic membrane. We have our synaptic cleft, and then we have our postsynaptic neuron. Okay, now the idea being that the action potential arrives at the synaptic knob, at the presynaptic membrane, and when the action potential arrives, calcium channels open. The influx of calcium causes the synaptic vesicles to fuse with the presynaptic membrane, releasing neurotransmitter by exocytosis into the synaptic cleft, whereupon it will diffuse to the postsynaptic membrane, or diffuse towards the postsynaptic membrane. The question is, what is happening there? So I've got a diagram here, and essentially what the diagram is showing is the composition. So it is showing the acetylcholine receptor. So in many cases, the standard neurotransmitter that's on our specification is acetylcholine, and the protein that it binds to is in fact a cation channel. That cation channel spends most of its time closed. However, when the acetylcholine binds to this receptor or to this protein, the channel opens, allowing the sodium. Remember that the resting potential is being maintained so that there's a high concentration of sodium outside the neuron. So when the neurotransmitter binds to the acetylcholine receptor, at that point the receptor, which itself is a cation channel as well, so it's got this kind of dual functionality. When it binds acetylcholine, the protein shape changes and it opens, allowing cations to diffuse down their concentration gradients. Now, in the case of sodium, it will allow the sodium to enter the postsynaptic neuron, causing depolarization to begin. Now, depending on how much neurotransmitter has been released, and again, remember that depends on how many action potentials have, or the frequency of the action potentials that are arriving at the presynaptic neuron. If there's a high frequency of those action potentials, in that case, there's going to be a lot of neurotransmitter being released into the synaptic cleft. That means on the postsynaptic neuron, there's going to be a lot of these sodium channels or neurotransmitter gated sodium channels being opened, causing a lot of influx of sodium into the postsynaptic neuron, and therefore enough sodium coming in to cause an action potential in the postsynaptic neuron. Okay, so let's look at the first question then. Are the receptors that the neurotransmitters bind to themselves membrane proteins? Yes, they are, as shown in this diagram. They are membrane proteins. They kind of have a lock and key kind of functionality. So as soon as these channels bind the neurotransmitter, the channels open, allowing the movement of cations down their concentration gradient. I think we're familiar with the idea that there's something called voltage-gated sodium channels or voltage-gated cation channels, and the calcium channel is an example of that, right? So, and we know that it's the changes in voltage that cause these channels to open or close. The idea being that while some channels are opened and closed by the voltage, other channels are opened and closed by the binding of certain molecules. So on the postsynaptic membrane, some of these channels are opened and closed by the binding or not of neurotransmitter. Okay, and in most cases at these synapses, at least at excitatory synapses, what's happening is that the neurotransmitter binds to the channel. The binding of the neurotransmitter to the channel causes the channel to open, allowing ions or cations to move down their concentration gradient. In the case of sodium, it means it's going to move from outside of the neuron to inside of the neuron, beginning the process of depolarization. Okay, now let us move on to the next part. Does that mean it's fair to say the neurotransmitters change the shape of the receptors or the sodium channel proteins? Yes and yes. So yes, they change the shape of the protein, absolutely. Do we call them receptors or do we call them sodium channels? I guess we call them sodium channels. That's their main job. The only thing is what's causing those channels to be opened or closed. All right, so we can call them receptors that are channels or ligand-gated channels. I prefer ligand, which is referring to the chemical or the neurotransmitter. These are ligand-gated channels. That's where I would fall on that one. Okay, right, next. Can one neurotransmitter cause an inhibitory response in one synapse and an excitatory response in another? Yes, they can, is the answer to that one. So I'll just move down here. So we already have an example of this, and again, acetylcholine is an example of a neurotransmitter that has, depending on the synapse or depending on where it is being released, it can have an excitatory function or an inhibitory function. Let's just remind ourselves about what an inhibitory synapse might look like and what it does. And that's what the diagram here is showing you, is that at an excitatory synapse, when the neurotransmitter is released and it binds to channels or cation channels on the postsynaptic membrane, when the neurotransmitter binds to its complementary ligand-gated channel, it causes the channel to open and allows the ions that will cause depolarization to enter the cell. So in an excitatory synapse, so it's a kind of, we'll think of it as a positive. In an excitatory synapse, the neurotransmitter binds to the ligand-gated channel and allows sodium to enter, causing depolarization. Okay, and what that means is the inside of the cell is becoming more positive as ions such as sodium are entering the cell. That's shown by this graph. So at a resting membrane potential we have -70. When the neurotransmitter is released, the potential inside the postsynaptic neuron increases to -60, beginning the depolarization process. Okay, so that's an excitatory synapse, and if you're not sure about any of this, then go back to resting potential and action potential and understand how that is working before you come back to this. Now, let's look at what happens at an inhibitory synapse. At an inhibitory synapse, same situation, got a presynaptic neuron, action potential is going to arrive, the influx of calcium will cause the neurotransmitter to be released, but in this case, the neurotransmitter binding to the channel binds to different channels, binds to different proteins, which they still have the capacity. So this is a different, this will be a different cell now. It can't be the same cell, because you can't have on the same cell the same neurotransmitter having opposite effects. Okay, so we'll look into how this works. But at one neuron, when the neurotransmitter released, it binds to, so one type of neuron has special types of sodium channels that when they bind the neurotransmitter, it allows the sodium into the cell. On a different neuron, in this type of cell, there may be a different type of channel protein that doesn't really involve the movement of sodium, but what it does is allows the movement of a different type of ion. So in this case, the same neurotransmitter is released. When it binds to the cation channel, it opens the cation channel to allow the influx of an ion, but that ion is not going to be sodium this time. It's going to be something that is going to act, in an opposite way to causing depolarization. So sodium being a positive ion, when it enters the cell, it causes depolarization. However, at an inhibitory synapse, when the neurotransmitter binds, it opens this ion channel, and this ion channel is going to allow the influx of ions, for example, chloride, Cl-, okay, that's a negative ion, it's an anion, and what that means is, instead of causing a depolarization of the cell, i.e. going from -70 to -60, it's not going to cause that. It's going to cause -70 to become -72, for example, and that is what we call hyperpolarization, and it's decreasing the chance that the cell is going to be able to reach the threshold potential and cause an action potential. So remember that when sodium comes into the cell, the potential inside the cell is, or the charge inside the cell is becoming more positive, and the more positive it gets, the more likely that we're going to reach the threshold potential. And at that point, we open the voltage gated sodium channels, we open enough of them to cause an action potential. But if you're causing hyperpolarization, you're decreasing the chance of the cell reaching the threshold potential. So in effect, you're delaying an action potential in these cells, and so the neurotransmitter is acting in an inhibitory capacity, it's reducing the chance that the postsynaptic neuron is going to undergo a depolarization, because you're decreasing the potential inside the cell to below -70 rather than increasing it to towards the threshold. So I hope that that kind of mechanistically makes sense. The key point being that, you know, let's kind of look a bit more closely at this. You can have a protein that is complementary to acetylcholine. So it's complementary to acetylcholine, but the rest of the protein, its job is to act as, let's say, a sodium channel. Okay. But you can have, on a different cell, you can have lots of proteins on the membrane. They are still complementary on their outward facing side to acetylcholine, but structurally, the rest of their structure is kind of optimized to allow the movement of not sodium, but chloride ions. Okay, so it is possible to have a protein that on one side is complementary to, so two proteins, so in this respect they are similar, but the job that they do in response to binding acetylcholine be different, i.e. one allows the movement of sodium in response to acetylcholine, the other one binds acetylcholine but allows the movement of chloride in response when it's opening. Okay, now the point being, you can't have these on the same neuron, because then acetylcholine is just going to be acting against itself, and that makes no sense. So let's look at an example of where this, how this works in practice. So when we're talking about neurons, and synapses, in this case, acetylcholine acts in an excitatory way. Okay, so when this happens, acetylcholine, so when we are talking about a synapse, at a synapse, acetylcholine acts in an excitatory way. So action potential arrives, chloride, the voltage gated calcium channels open, calcium enters the presynaptic neuron, synaptic vesicles fuse with the presynaptic membrane, neurotransmitter released, this being acetylcholine. Acetylcholine binds to the ligand gated sodium channels, and when acetylcholine binds, it allows the entry of sodium into the neuron, beginning the process of depolarization. And if there's enough neurotransmitter, there will be an action potential, because the size of the depolarization will reach the threshold potential. That's at a neuron or a synapse. However, when we are talking, acetylcholine also is used as a neurotransmitter in a different situation, that being the sinoatrial node of the heart. Okay, and at the sinoatrial node, remember the heart rate is kind of regulated by the sinoatrial node, but the heart rate can be modified via the nervous system. So there's two nerves that connect to the sinoatrial node, and they modify the rate of depolarization of the sinoatrial node, thereby affecting heart rate. Okay, now how does that happen? Again, so we're talking about synapses here, and the sinoatrial node, the cells of the sinoatrial node are responsive to neurotransmitter released by the nerves that are connected ultimately to the cardiovascular control center, in the medulla oblongata. Okay, so connected to the brain via these nerves. Now, those nerves, there's two of them, because sometimes you need to increase the rate of the SAN because you want to increase the heart rate, because oxygen supply needs to be increased, carbon dioxide removal needs to be increased, and so on. But when the exercise is done, or in the situations where we no longer need those increased rates of supply, in that case we need to be able to also reduce the heart rate, and this is how we maintain proper control. So not only do we need to be able to increase, we need to be able to decrease, and the way we do that is by having excitatory synapses and inhibitory synapses. So at the excitatory synapse, the neurotransmitter that is released is noradrenaline. Okay, and noradrenaline acts in an excitatory way. It increases the rate of depolarization of the sinoatrial node. Acetylcholine, so we should remind ourselves, and this is the accelerator nerve of the parasympathetic nervous system. Okay, acceleratory nerve of the sympathetic nervous system. So we can call it the sympathetic nerve, or the accelerator nerve. Okay, right, so there we have that. Now the other nerve is of the parasympathetic. Or it's called the vagus nerve. Okay, and when the impulses move down the parasympathetic nerve, the neurotransmitter that's released in this case is acetylcholine. And when acetylcholine is released here, when the nerve impulses, so the situation is that the heart rate needs to be reduced, then the impulses go from the cardiovascular control center, the impulses move down the parasympathetic nerve, or otherwise known as the vagus nerve, and when the action potential gets to the end of these neurons, the neurotransmitter that's released is acetylcholine. And when acetylcholine binds to the cells of the sinoatrial node, it causes the rate of depolarization to decrease. Most likely because it's causing the levels of depolarization to go down, because the neurotransmitter is acting in an inhibitory manner, reducing the depolarization of the sinoatrial node cells, thus reducing the heart rate, the rate of contraction of the heart, and effecting the appropriate response as kind of controlled by the cardiovascular control center. And there we have it. So let's just go back and see whether we've addressed that. Can one neurotransmitter cause an inhibitory response in one synapse and an excitatory response in another? Yes, they can. It doesn't make any sense because the neurotransmitter only binds to receptors which are complementary to its shape. That is true, but remember, just because they're complementary on their outward face doesn't mean that the rest of their structure can't be different to allow a different response to the same signal. Okay, and I hope that has made sense. Some person, others listening, if you have other questions, that allows us to touch on biology topics in this way and allows us to get a little bit more deep into the biological concepts, I'd be happy to do another video like this that hopefully helps you understand biology a little bit better, and just take you to the next stage of your understanding. Again, I'm sure you've understood a few things today, but that's just raised more questions, and that's a good thing. That's moving forward as a scientist. It's just always about the next question, really. Okay, good luck, guys.